Hi, everyone, and welcome to a special Trial Site News interview. I'm Brandon, your host. And today we'll be chatting with Dr. Robert Malone, who has been interviewed by Trial Site News in the past, as well as contributed numerous articles to our website. He is a scientist, vaccinologist, author of the book Lies My Government Told Me, and currently authors a very popular Substack titled Who is Robert Malone? And Dr. Malone, thanks for joining us today.
Thanks for having me on, Brandon, and good to be back talking to the Trial Site News community.
Thank you. Let's talk about the latest COVID booster shots. In one of your latest Substack essays, you wrote that the current booster approved for emergency use is actually obsolete. Can you explain why?
Yeah, I'll be glad to. I'm going to walk you through, try to be quick. These boosters, quote unquote, that were rolled out under emergency use authorization, even though we no longer have an emergency. Remember, the emergency concluded last May. I think it was May 11th. But the FDA unilaterally decided they would extend that for another, I think, 90 days or 6 months. It must have been 6 months. And we're just coming up to the closure on that window that the FDA carved out for itself. And during that window, they, on the basis of the recommendation from the VRBAC, the Vaccine-Related Biologics Advisory Committee at the FDA, authorized purchase from the manufacturers of these vaccines, Novavax, Pfizer, BioNTech, and Moderna, authorized the pre-purchase, basically, agreement for boosters for the fall that would be based on the Kraken variant. You all remember the Kraken, very scary, especially if you were on the crew of the Pirates of the Caribbean series, and was associated with mass deaths, bodies in freezer vans, and large mass graves. No, none of that happened. But it was a very scary name, and the FDA VRBAC thought that that was going to be the circulating variant come fall. And so, like they do with influenza, they made a forward-looking prediction, and that turned out to be wrong. By the time the boosters were manufactured and jammed through the FDA authorization process under EUA, Kraken had basically become extinct in the face of competition of Pyrrhus and Eris, E-R-I-S, as I recall, as the two dominant variants, particularly Eris. And Eris variant is in a separate phylogenetic branch of the evolutionary tree of Omicron derivatives. And so they built a vaccine against Kraken, which is in one branch of the XBB Omicron lineage. And by the time the fall rolled around, it had been outcompeted by this Eris variant, which is another branch and has different mutations that are even more resistant to to antibody neutralization. And so the FDA, in the face of this, in order to get this out to the public, authorized the use of mouse serum samples generated by vaccinating, I think it was 11 mice, and then pooled as a material to be tested under virus neutralization assays, which is a method to detect in cell culture whether or not antibodies will block the ability of a virus strain to infect a target cell in basically petri dishes. They're not petri dishes, but the metaphor works. And so on the basis of that, which we were never shown the data, the FDA asserted that there was enough cross-reactivity between these Kraken-based vaccines expressing the Kraken spike and this new evolved spike of Eris that they would still be effective. They didn't tell us effective for what, because it's already been well established that these vaccines do not prevent infection, replication, or spread. In fact, against Omicron, the prior rounds of vaccines had between 0 and about 35% effectiveness in preventing infection. And so the FDA has fallen back over time onto a new endpoint. Since we're talking trial site news, we can use industry language. So a new endpoint for this authorization, which was reduction in disease and death, which was not one of the original endpoints, and which was not tested. But that's how this has gone. So now what's happened is, even in this short interval since these, quote, boosters designed against Kraken have been deployed, remembering that we only have, according to PBS, between 2 and 3% uptake in children and 7% uptake in adults. But during that time frame, while these are being marketed, frankly, to the general population, we've had the rise of a new variant called HV.1. Internet virologists have noted that this sounds an awful lot like HIV, but it has nothing to do with HIV. It's just the nomenclature.
Okay.
So this HV.1 is basically a further evolution of Eris. So it's in the same lineage branch, which is very different from the Kraken branch. But it has a couple of very intriguing changes. It is highly, highly resistant to antibody neutralization. And it has also acquired, in a strange way, it's difficult to discern this, maybe it's convergent evolution, but it has acquired one of the evolutionary modifications that were present in Delta. Now, what made Delta so pathogenic appears to have been its predisposition to infecting deep lung. And in other respiratory viruses, this has been known to be a problem and associated with more severe disease. This HV.1 does not have that characteristic. That is not what it acquired from Delta, but it's rather a receptor binding domain mutation that makes it even more resistant to antibody neutralization. So now what we have is a variant that seems to be completely resistant to the known neutralizing antibodies, which means that our last line of protection is cellular immunity and innate immunity. The boosting, quote unquote, with these mismatched Kraken-based boosters has no data indicating that they will elicit a neutralizing antibody response that will block infection by HV.1. So hence my assertion that currently we have no data to support the continued use of these boosters, which absolutely do have adverse events associated with them. They're not completely benign. We can argue about back and forth about how severe those adverse events are, how frequent they are. But it's not debatable that these adverse events are occurring at a significant level. The FDA asserts that these are rare adverse events, but they don't quantify what rare consists of. Is it 1 in 1,000? Do they consider rare a adverse event that happens at greater than 1 in 100? We don't have any information about what does rare adverse event mean to the FDA currently, nor do we have any information about what the FDA considers safe and effective, which is their mantra all the way through this, that these products are safe and effective. Effective in what way? Because we have no indication that they are able to reduce the risk of infection from this current variant. And we have no data about whether or not they are able to reduce the risk of disease or death. They certainly don't prevent disease or death. And And so what we're left with is ongoing propaganda for truly an obsolete product that the virus has evolved to escape, and for which we have no data at this point about what positive benefit is associated with this product. We do have abundant data on what the adverse event profile is of these products, and those data continue to grow, and the profile of adverse event risks continues to expand. And now we have this new information about what I assert is adulteration. The inclusion of short DNA fragments that are not purified away from the RNA, but are co-formulated with the lipid nanoparticles and delivered to cells and tissues of those that receive the products. And we know from prior work that short DNA fragments are genotoxic or recombogenic. They have significant risk of causing insertional mutations in cells that receive these short DNAs. But the FDA has no data about what the risks are associated with that, and once again is relying on blank denials of risk and assertions that since over 1 billion doses have been administered, and the FDA concludes from that history that these are, quote, safe and effective, that they have no concern about these short DNA fragment contaminations and their potential clinical implications. So that's kind of where we stand right now, is HV.1, the new one that is taking over globally in the United States and displacing Eris, which long ago displaced Kraken, is mismatched, and probably the current, or let's say it this way, we have no data indicating that the current boosters have any effectiveness against HV.1. And we do now have data that apparently European Medicines Agency, Paul Ehrlich Institute, Health Canada, and FDA seem to have been unaware of, particularly in the case of Pfizer, of significant contamination with short DNA fragments that are being delivered. And within those are short DNA fragments, including a highly active genetic switch, a promoter enhancer, derived from simian virus 40. Now, this is not the whole SV40. This is just a fragment of its DNA. But it happens to be one of the most highly active genetic switches known to molecular biology. And that potentially can integrate. And if so, prior FDA guidance in the case of DNA vaccines would indicate that that is a high risk and should have been known and evaluated. But it appears that those that prepared the Pfizer BioNTech regulatory submissions, when they provided the plasmid map of the DNA that was going to be used under the new manufacturing process, so this is process 2, they did not include in that plasmid map identification of these simian virus 40 derived sequences in the plasmid, which is very odd because any standard plasmid mapping program, circular DNA produced in bacteria being a plasmid, that is the template for manufacturing the RNA, any standard program would have flagged those SV40 sequences. So the appearance is that so the inference, we don't have the receipts. The inference is that since virtually all modern plasmid mapping programs would have flagged a sequence, the appearance is that such a flag must have been deleted before the documentation was provided to the FDA, and then and Health Canada and European Medicines Agency, and apparently EMA, Health Canada, and FDA did not take the sequence loaded into their own mapping program to cross-check what had been submitted by the sponsor. So that seems to indicate a failure of rigorous diligence on the part of the regulators at best. And part of that failure now in subsequent research is the reveal that this plasmid being used for manufacturing by Pfizer has a long open reading frame. This is a way of saying it could produce a protein. In that, which was previously undisclosed, in addition to the SV40 sequence, which is relatively homologous to various spider silk proteins. So this is another one where the FDA, EMA, and Health Canada appeared to have been asleep at the switch and not doing their job. And we can only speculate about why that might have been the case. And personally, I prefer to avoid speculation about what's going on in people's minds, because I still am not a mind reader, despite my 64 years.
So I think you've already answered, I believe, my next two questions. So I'll maybe bring them together and see if you want to comment any more. But you mentioned the Kraken, you mentioned Eris, and HV.1. Basically, the vaccine makers can't keep up with this virus as it evolves. This is a race where, it seems the vaccine makers will never take the lead. Is it even possible for them to ever catch up with the virus as it evolves? And secondly, I think you've already answered this, and I'll just loop this in as well. Are the boosters actually helping anyone at this point? For example, the elderly or someone in a high-risk population? Or is there any reason at this point for anyone to take them?
So the first point is the most fundamental, because this is what has been used to justify jamming this mRNA or modified mRNA technology platform through the regulatory process. And it's the reason why the Nobel Prize was granted to Carrico and Weissman, explicitly as stated by the Nobel Committee. Number one, to help motivate people to take these products, remembering that the Karolinska, which chooses who gets the prize, has financial ties to both Moderna and Pfizer, surprisingly, I'm sure. And they're not trivial. But the justification has been that the identification of pseudouridine incorporation has enabled this very rapid vaccine development using this platform. And it was believed to be necessary to do exactly what you're highlighting, to be able to get out ahead of either engineered pathogens produced by the bad guys, whoever those are, garage biologists or whatever the entity is. So the thesis is that this tech can allow manufacturers to very rapidly produce products that would enable vaccine deployment timelines to be shortened to the point where they could respond in real time to evolving viruses or newly engineered pathogens. The evidence, as I see it, supporting the timeline for manufacture and deployment is not so much the creation and engineering of the candidate product, but rather the willingness to not impose standard regulatory processes. I'm choosing my words. That it's the willingness to not impose normal regulatory processes for toxicology, genotoxicity, reproductive toxicity, et cetera, all these things, and instability, et cetera, that the Trial Site News readership is well familiar with as taking considerable time in being able to deploy a new candidate product into initial human clinical trials and then subsequently as a licensed product or a market authorized product. And it was the suspension of those that allowed all this to happen quickly. And the proof of the pudding for that is that, for instance, the Russian vaccine products, which also were produced by dropping a lot of regulatory and clinical trials norms and non-clinical data norms, were produced at approximately the same timeline without being a genetic technology. So in this case, the thesis is that this new technology platform enables rapid response. And yet here we have an example where all of those rules have been suspended. Emergency use authorization has still been authorized. They actually didn't do any human clinical testing. They relied on mouse vaccination, which any vaccinologist will tell you, the saying is mice lie, monkeys mislead, and the only thing that predicts protection in humans is protection in humans. The mouse immune system is not human immune system. It is a long way away, especially these inbred mouse lines. But FDA parked all of their normal processes, jammed this through, and they still couldn't do it in time. So that really proves your point that this virus, which is evolving more rapidly than even a normal beta coronavirus would, in the clinical and virologic experience of beta coronaviruses that are one of our common colds. And one of the theses for why this rapid evolution is being driven has to do with the deployment of leaky vaccines. But regardless, clearly they can't get ahead of the curve, even with all of this expedited process, even with the mRNA platform, even with not having good manufacturing controls and lot release controls, because that's why we end up with these DNA fragments not having been detected, is because they didn't do good lot release. And then there is this bubbling up concern again about vaccine enhanced disease, and in particular whether in Delta, in the interim between dose 1 and dose 2, those that had received dose 1 actually had proportionately more deaths. And that's a current topic of investigation. That's out on the edge, unproven, but there's statistical analyses that suggest that might be the case. So the whole, antibody-dependent enhancement is one form of vaccine-enhanced disease, but that topic now is coming back up to fore. So we've got a whole bunch of things that are converging on maybe it wasn't such a good idea to jam this thing through, and we really didn't get any benefit in the long run from having done so.
Okay. So your latest article on Substack covers the Jim Jordan release of documents surrounding this Orwellian group of partners called the Election Integrity Partnership. Can you tell us more about this?
So for anybody that wants to dive into the primary documents supporting this, because I advocate that you don't rely on my assessment, make your own analysis and your own decisions about what this all means. But Jim Jordan has decided to release a report. Apparently, it appears out of frustration with slow walking and foot dragging going on in some of his congressional committee members' offices. So he has unilaterally dropped this report titled Weaponization of Disinformation, Pseudo-Experts and Bureaucrats, How the Federal Government Partnered with Universities to Censor Americans' Political Speech. And what it documents is that now many of us are coming to know CISA, this shadowy agency that has basically been coordinating all of the propaganda and censorship and defamation and character assassination things that have been going on since before the last presidential election. And what CISA and another federal agency that goes by the acronym GEC did was set up a public-private partnership with a consortium called the Election Integrity Partnership under the umbrella of the Atlantic Council. And for those of you that subscribe to the deep state thesis, Atlantic Council is like a home base for the deep state. So, and you can look up the Atlantic Council, and we have links here and make your own decisions about that. But Election Integrity Partnership was set up with the Stanford Internet Observatory to monitor for election disinformation in the prior presidential election. And then as that election came to closure, it was transformed into a focus on vaccine-related disinformation and misinformation and malinformation, all of which are defined by the Department of Homeland Security as domestic terrorism, just to put a pin on that one. And so what happens is the federal government flags things that it believes are different from its approved narrative or that of the WHO, feeds those into this election integrity partnership, which then has a bunch of resources in a stepwise fashion to work cooperatively to identify who's saying these things, where they're saying it, how it's getting amplified, who's retweeting it or reposting it, and flagging all those as posts, information, social media activists that they then feed to the big tech enterprises like Google, Twitter, Facebook, TikTok, and then advocates that, and YouTube, of course, advocates that those people and those bits of information be censored or deplatformed. So that's been the biosphere here. And this only dropped 2 days ago. And what Jill Malone, Dr. Jill Malone, my partner and everything, did with this particular essay, I edited it, but she was the primary author, was to dive deeply into those documents that were cited by Jim Jordan's report, and found that among those that had been so targeted was Dr. Robert Malone, but many others also. But in particular, as this transition from election integrity went towards disinformation about the vaccine products, and what the offending interaction that caused me to be flagged as a repeat offender and identified as an anti-vaxxer, which is an odd thing to call me, somebody that spent much of his career developing vaccines.
Right.
I'm just an objector to what's been done with this vaccine or set of vaccines in this context. But they flagged me as a repeat offender, misinformation spreader, anti-vaxxer, and worst of all, conservative. So I was flagged for being conservative among the other things. Right. And then they promoted a series of actions on the basis, the specific inciting event, bless their heart, they identified, which was a interview that Tucker Carlson did with me back in 2021, in which I asserted Tucker was querying me about some new Japanese data that your viewership may remember, having to do with myocarditis and the incidence of myocarditis in young males. And he was asking me about those data and why we aren't seeing this in the United States. And I asserted that the FDA and HHS and CDC were hiding data from the public. So that was what triggered all this. Now we know, because of Freedom of Information actions, that in fact they were hiding these data, these specific data, from the public. So what I said was absolutely true. And I was just corresponding with Tucker about this. I flagged him. I have his personal contact information. I flagged him and say, hey, Tucker, you should know about this because you were the trigger. And he said, he finds this whole thing amazing because everything that I had said and others has been verified as true. Yet we were subjected to this censorship, defamation, gaslighting, and character assassination on behalf of the federal government by Stanford, the Atlantic Council, and this consortium. But this is not about me. This is about a given case study that shows how this whole ecosystem has been run, that's prevented scientists and medical professionals and regulatory professionals from discussing their reservations about the positions taken by the government as regards to these products. And this is clearly a smoking gun, one of many that now have come out.
Dr. Robert Malone, thank you so much for joining us today.
My pleasure. And I look forward to the next chance for us to chat and for me to contribute to Trial Site News. Just to put a pin in this one, it was the essay in Trial Site News where I discussed the bioethics of what was being done that really put me on this pathway of, let's say, coming out of the closet as a conscientious objector or whistleblower, depending on how you want to pitch it, that has led to all of the stuff that has enveloped me over the last 3 years. And I thank Trial Site News for its courage and willingness to allow me to speak about the bioethics of what was going on at a time when that was a sensitive topic, and really alerting much of the nation and the world about the harms and risks of the path that they'd undertaken.
Well, thank you again, and please visit trialsitenews.com for unbiased news and opinion articles on the latest in scientific research.